Hello, my name is Michelle Monje. I am a professor of neurology at Stanford University and a pediatric neuro-oncologist. Today, I will tell you a story about interactions between neurons and the glial cells that form the myelin sheath, how these interactions promote adaptive changes in myelin that contributes to healthy cognitive function, and how these powerful neuron-glial interactions are subverted in the context of glial malignancies. Glial malignancies occur in a predictable neuroanatomical place at particular ages, so that really suggests dysregulation of a developmental process. And this pattern of glioma incidence maps well onto the spatiotemporal pattern of developmental myelination, such that at a time when there is a discrete wave of myelination in the ventral brainstem, during mid-childhood, this is when the universally lethal diffuse intrinsic pontine glioma tends to occur. And similarly, when there's an active wave of myelination in the neocortex in young adulthood and adolescence, this is when pediatric hemispheric high-grade gliomas tend to occur. And these observations are concordant with discoveries from my laboratory and from many others that many forms of glioma, including those traditionally thought of as astrocytomas, may in fact arrive from precursor cells in the oligodendroglial lineage, either oligodendroglial precursor cells or earlier neural stem cells that frequently go down the oligodendroglial differentiation pathway. And so we may glean really important clues about gliomagenesis by better understanding what normally regulates the process of gliogenesis. And this poses a really basic question about what regulates oligodendrocyte precursor cell proliferation and the generation of myelin-forming oligodendrocytes. One idea that's been in the literature for some time, but that remained hotly debated until recently, is the idea that neurons themselves may regulate the extent to which their axons are myelinated, that myelination may be activity and experience dependent. While supported by beautiful in vitro and correlational work, this idea remained controversial, in part because there are also clearly activity-independent modes of myelination. And so for several years, my laboratory has been working to understand if neuroglial interactions regulate normal and malignant oligodendroglial precursor cells. To test interactions between neurons and glial cells, we've used optogenetic stimulation of projection neurons in the motor planning area of cortex, which elicits complex motor output. So we know that we've successfully stimulated the premotor circuit. And then we can ask straightforward questions about how other cell types in the circuit respond to changes in neuronal activity. We discovered that neuronal activity results in rapid and robust proliferation of oligodendrocyte precursor cells in a circuit-specific and neuronal subtype-specific manner. Fate mapping these proliferating glial precursors over time, we found that new oligodendrocytes are generated that subtly change the myelinated ultrastructure of the circuit in a way we would predict would alter circuit dynamics and perhaps influence function. And indeed, we discovered that motor functional behavior improved in these mice over time, and that this improvement depended upon the generation of new oligodendrocytes. The newly generated oligodendrocytes in the healthy brain myelinate naked axons or regions of discontinuously myelinated axons, as well as remodel existing myelin internodes. Overall, these relatively small changes fine-tune circuit dynamics to promote coordinated circuit function. The mechanisms that mediate this plasticity of myelin have been and remain a major focus in my laboratory and many others. We know from the early work of Dwight Burgles and others that bona fide synapses form between neurons and oligodendrocyte precursor cells. And we've discovered through monosynaptic viral tracing experiments that OPCs receive broad synaptic inputs, but how these cells integrate this information about circuit activity remains a really important open question. We've also discovered that one element of this plasticity mechanism involves activity-regulated BDNF signaling to its receptor TREC-B on oligodendrocyte precursor cells. And this discovery gave us a molecular handle to specifically block activity-regulated myelination and ask what role myelin plasticity plays in normal function. This approach has uncovered a range of important roles for myelin plasticity in various cognitive processes, including attention, memory, and learning. But how does myelin plasticity contribute to disease when the glial precursors are malignant rather than normal? Could these neuron-glial interactions be subverted in glioma? Using that same premotor optogenetic paradigm, but this time in the context of a pediatric glioblastoma taken from one of my patients and placed in the mouse cortex, we found that neuronal activity similarly increases the proliferation of malignant glioma as it does its normal counterpart, and that this results in an increase in tumor burden, specifically within the stimulated circuit, demonstrating that brain activity can drive brain cancer growth. Similarly, optogenetic stimulation of the optic nerve in a genetically engineered mouse model of optic pathway glioma also increases tumor growth. Not only does neuronal activity promote glioma growth, but we found that decreasing neuronal activity regulates tumor initiation and maintenance. In this optic pathway glioma model, there is reliable generation of optic nerve tumors around 9 weeks of age. And so we can modulate then neural circuit activity simply by placing mice in complete darkness to decrease visual experience around the time of tumor initiation. And when we do that at 9 weeks of age, at the time that tumors form, or just after at 12 weeks of age, we find that far fewer and much smaller tumors form compared to litter-mate controls raised with normal visual experience, with normal optic nerve activity. Furthermore, dark rearing these mice prior to tumor onset blocks tumor initiation entirely, despite the cell intrinsic vulnerability to the tumor predisposing genetic background of these mice. So what are the mechanisms that mediate these really powerful interactions? Well, hypothesizing that paracrine factors may mediate these powerful effects of neuronal activity on glioma growth, we performed a secreted factor screen by collecting conditioned medium from either cortical or retinal explants that were either optogenetically stimulated, spontaneously active, or in which neuronal activity was silenced using the voltage-gated sodium channel blocker tetrodotoxin. And then when we place this explant conditioned medium onto cultures of glioma cells, we find that there's an activity dose-dependent increase in glioma proliferation that's blocked when action potentials have been silenced with tetrodotoxin. Now, as this audience knows well, gliomas are a group of clinically and molecularly distinct diseases. But we found that this response to activity-regulated secreted factors is conserved across a wide range of glial malignancies, including pediatric histone wild-type hemispheric high-grade glioma, histone mutant diffuse midline glioma, or DIPG, IDH wild-type adult glioblastoma, IDH mutant anaplastic oligodendroglioma, and NF1-associated low-grade optic glioma. So what's in that conditioned medium? Well, performing biochemical and proteomic analyses followed by sufficiency and necessity testing, we found that there were two key activity-regulated mechanisms we could identify using this experimental setup. Not surprisingly, because it plays such a critical role in normal neuron-glial interactions, we found that brain-derived neurotrophic factor, BDNF, is one important activity-regulated mitogen for gliomas. But really unexpectedly, we found a novel shed form of neuroligin-3. Neuroligin-3 is a well-studied postsynaptic adhesion molecule. It plays multiple roles at the synapse, regulating synaptic strength of both excitatory and also inhibitory synapses between neurons. It was not known, however, to be a mitogen in any context, and actually it was not even known to be secreted. But we found that neuroligin-3 is cleaved at the membrane in a strictly activity-dependent manner through the enzymatic activity of a metalloprotease called ADAM10. So the next basic question was, what cell type is shedding neuroligin 3? While neurons are, of course, postsynaptic cells, oligodendrocyte precursor cells are also a postsynaptic cell type and actually express very high levels of neuroligin 3. Through cell type specific conditional deletion of neuroligin 3, we discovered that not only neurons, but importantly OPCs shed neuroligin 3 in an activity dependent manner, raising important questions about the role that neuroligin 3 may play in normal myelin biology and placing the oligodendrocyte precursor cell in the tumor microenvironment together with neurons. Our next set of questions focused on the relative importance of this mechanism for tumor progression, given the many cell intrinsic and multiple microenvironmental mechanisms that could influence glioma progression. So to test the relative contribution of neuroligin 3, we xenografted patient-derived brain cancer cells into the environment of either the neuroligin 3 wild type or neuroligin 3 knockout brain. Surprisingly, we found a complete stagnation of tumor growth in the absence of neuroligin 3. This dependency on microenvironmental neuroligin 3 extended to multiple different forms of glioma, from pontine glioma to pediatric and adult glioblastoma. But this did not extend to a patient-derived model of breast cancer brain metastasis, suggesting that while neuroligin 3 is a key factor regulating the growth of glial malignancies, it may not play a central role in all forms of brain cancer. So we next return to that NF1-associated optic pathway glioma model, and this is work that we did in collaboration with David Gutman's lab at Washington University. We simply crossed NF1 optic pathway glioma mice to neuroligin 3 knockout mice, hypothesizing that neuroligin 3 may be playing a similar role in low-grade glioma progression, as we have found that it plays in high-grade glioma progression. And I'll mention that, one of the reasons we thought neuroligin 3 might be an important mechanistic factor in optic pathway associated low-grade glioma as well is because in the NF1 optic nerve, there is aberrantly increased neuroligin 3 shedding, suggesting that part of the tumor predisposition to optic gliomas could be accounted for by this aberrant neuroligin 3 shedding in the NF1 optic nerve. When we crossed these mice, we found that even with normal visual experience and normal optic pathway activity, in the absence of neuroligin 3 in the optic nerve, there are far fewer and much smaller tumors that form compared to neuroligin 3 wild type littermate controls. So I've just told you that neuroligin 3 is a really interesting therapeutic target, and also that ADAM10 is the enzyme that mediates its cleavage and release into the tumor microenvironment. So we tested the idea that pharmacological inhibition of ADAM10 may phenocopy neuroligin 3 loss in the tumor microenvironment, and found that blocking neuroligin 3 shedding indeed robustly slows tumor growth, both in high-grade glioma as well as in optic pathway low-grade gliomas. But why is neuroligin 3 such a powerful molecule in glioma progression? Well, we discovered that when neuroligin 3 binds to the tumor cell, it stimulates numerous oncogenic signaling pathways. And this helps to explain its sufficiency in promoting glioma growth, but it really doesn't explain that unexpected dependency. So we dug deeper and examined gene expression changes attributable to neuroligin 3 binding. And what we found was that there is a feed-forward effect of neuroligin 3 on a number of synapse-associated genes. There's a feed-forward effect of neuroligin 3 on its own expression, together with upregulation of the BDNF receptor TrkB, as well as a number of other glutamate receptor subunits and other synapse-associated structural proteins. This raised the kind of crazy idea that, like there are synapses between neurons and normal glial precursor cells, there may also be synapses between neurons and glioma cells. When we explored this possibility using immunoelectron microscopy to unambiguously identify glioma processes by immunogold labeling, we found these clear synaptic structures between presynaptic neurons and postsynaptic glioma cells. You can see here this postsynaptic density on the glioma cell, a very clear synaptic cleft, and presynaptic neurons containing synaptic vesicles. testing the idea that perhaps a fundamental role of neuroligin-3 in glioma pathophysiology is to promote the formation of these synapses. We indeed found far fewer structural synapses form between neurons and glioma cells in the absence of neuroligin-3 from the tumor microenvironment. But are these neuron to glioma synapses functional, or are these simply a shadow of the cell type from which we believe that many of these gliomas emerge? To answer this question, we performed whole cell patch clamp electrophysiology, recording from glioma cells xenografted to the CA1 region of the experimentally tractable hippocampal circuit, while stimulating the axon afferents into CA1. We found excitatory postsynaptic currents in a subpopulation of glioma cells within each patient-derived tumor. These EPSCs depend upon action potentials. They're blocked by the voltage-gated sodium channel blocker tetrodotoxin. And these synaptic currents exhibit multiple electrophysiological characteristics of bona fide synapses, including paired pulse facilitation and single vesicle events called mini EPSCs. The neuron to glioma synapses that we first identified are glutamatergic, calcium permeable AMPA receptor dependent synapses. In the healthy brain and between neurons, there is adaptive plasticity of synaptic strength and synaptic connectivity. And we wondered whether glioma cells may be exhibiting similar mechanisms of adaptive plasticity in response to activity regulated factors. One way that healthy neurons do this is through coincidence detection using NMDA receptor mediated mechanisms. Now, interestingly, glioma cells do not express NMDA receptors to in any appreciable degree. And so we wondered whether an alternative mechanism, such as brain-derived neurotrophic factor, might mediate this sort of malignant synaptic plasticity. And so we tested this idea by perfusing BDNF over our glioma cells while performing patch clamp electrophysiology, and found that indeed the glutamate evoked currents in the glioma cells have a much larger amplitude in the presence of BDNF than in the absence, and that this effect is lost if we CRISPR delete the BDNF receptor and TREC2 or TREC-B from the glioma cells. In healthy cells, one way that BDNF and other mechanisms promote a sort of adaptive increase in synaptic strength is through trafficking of AMPA receptors to the synaptic membrane. And so we thought that might be going on in the glioma cells as well, and tested this using multiple mechanisms. I'm showing you here cell surface biotinylation studies showing that in the presence of BDNF, that there is an increase in AMPA receptor subunits inserted into the glioma cell membrane. We've also identified a second kind of neuron to glioma synapse. These are particularly prominent in H3K27M mutant diffuse midline glioma cells. And so while the AMPA receptor mediated synapses are present both in adult GBM and in H3K27M mutant DMG, we find that GABA mediated synapses are much more prominent in H3K27M DMG cells. Testing for these GABAergic synapses in H3K27M DMG cells, we find that there is a GABA evoked current that is blocked in the presence of tetrodotoxin, and that there is a real difference between both different patient-derived models in terms of the strength, the amplitude of these GABA-evoked currents. So here's two different H3K27M DMG lines in blue and in black. They both exhibit these currents in response to GABA, but they're very different in strength. In contrast, IDH wild type adult GBM cells really do not exhibit much electrophysiological response when exposed to GABA. Now, in healthy neurons, GABA is a hyperpolarizing inhibitory neurotransmitter. But in neural stem and precursor cells and in immature neurons, instead, GABA can be excitatory. It can be depolarizing. And that's because of a difference in the chloride concentration intracellularly of immature cells. And we find that the glioma cells, just like immature precursors, exhibit depolarizing current in response to GABA. Now, in another subpopulation of cells, rather than these synaptic currents, we instead saw a very prolonged current. The synaptic currents are on the millisecond timescale. These prolonged currents are seconds long. Interestingly, these prolonged currents scale with field potential, meaning that the more neurons are active, the larger and longer these currents are. And what we determined that these prolonged currents represent are potassium-evoked currents. They can be evoked by changes in extracellular potassium, as occurs with action potentials naturally, and can be blocked with barium, which blocks potassium channels. Now, interestingly, when we dye fill the cells after recording from them, which we do so we can go back and confirm that the cell we were recording from was indeed a malignant cell, what we found when we dye filled these cells that exhibit these prolonged currents was that the biocytin dye diffused not just to one glioma cell, but actually to a network of glioma cells. And this evoked for us the really elegant work by Frank Winkler's lab, demonstrating that glioma cells can form an interconnected network coupled by gap junctions. We wondered whether this gap junctional coupling may also be electrically coupling these cells and amplifying some of these activity-dependent currents. And so we blocked gap junctions and found that indeed this decreases the amplitude of these potassium-evoked currents. And so to summarize, we have discovered that gliomas are integrating into neural circuits. The cancer cells integrate synaptically through AMPA receptor-dependent synapses in many forms of high-grade glioma, as well as through GABAergic synapses in H3K27M mutant diffuse midline gliomas. These synaptic connections are elaborated and reinforced through mechanisms of malignant synaptic plasticity. These tumors are also integrating electrically through potassium-evoked currents that are then amplified in a gap junction coupled network between glioma cells. Given that glioma cells exhibit multiple mechanisms of depolarizing currents, we hypothesized that membrane depolarization itself may be fundamental to brain cancer growth. And this would make sense given that membrane depolarization regulates normal neural stem and precursor cell proliferation during brain development. So to test for membrane voltage-dependent mechanisms of glioma proliferation, we again turned to optogenetics, but this time placing the light-sensitive ion channel, the light-sensitive opsin in glioma cells rather than in microenvironmental neurons in order to optogenetically depolarize the xenografted glioma. We found that indeed membrane depolarization alone drives glioma proliferation through voltage-sensitive signaling mechanisms that we're now working hard to identify. Conversely, blocking glutamatergic signaling between neurons and glioma cells, either pharmacologically or genetically by expressing a dominant negative AMPA receptor subunit, GluA2, in the glioma cells prior to xenografting, starkly decreases glioma growth. We can visualize glioma currents using genetically encoded calcium indicators expressed in the cancer cells. And this underscores the startling realization that this cancer is an electrically active tissue. This fundamental property of glioma had really not been appreciated until now, and it is central to disease progression. We must understand the neuroscience of this malignant circuitry at the systems, cellular, and molecular levels. The challenge is before us to begin to understand mechanisms of malignant circuit assembly, plasticity, and evolution over the disease course, voltage-sensitive mechanisms of proliferation, and to identify neurophysiological targets in this cancer that have so far gone unappreciated. Already, we're beginning to see potential ways to intervene therapeutically on neuron-glioma interactions, either by blocking neuroligin 3 shedding or binding, blocking glutamatergic signaling to the glioma cell, blocking BDNF to TREC-B signaling between neurons and glioma cells. The mechanistic parallels that are evident between healthy neuron-glial and cancerous neuron glioma interactions really underscores the extent to which these tumors are hijacking mechanisms of normal neural development and plasticity, and demand that we begin to approach these cancers from a neuroscience perspective. I want to end with the idea that the interaction between neurons and glioma cells is not unidimensional. Just as neuronal activity drives glioma cell progression through activity-dependent paracrine factor secretion and neuron-glioma synaptic communication, so too do the glioma cells increase neuronal excitability and activity, creating a vicious cycle between neurons and glioma cells. And I'll end with the idea that neuroscience is important not just for brain cancer, but really quite broadly in cancer in general. There are electrochemical neuron cancer, interactions that we've discussed now in glioma. Doug Hanahan's group has found these in brain metastases as well. Really important paracrine activity-dependent signaling between peripheral nerves and peripheral cancers, just as we observe in brain cancers. And also really important and very immune modulatory interactions between peripheral cancers and the nervous system. There are many people to thank for this work. I want to especially highlight Hamza Venkatesh, who was a brilliant cancer biology student and later a postdoctoral fellow in my lab, who led much of the work that I just showed you. She's now an independent investigator at Dana-Farber Cancer Institute and Brigham and Women's Hospital. Yuen Pan, who has just completed her postdoctoral fellowship in my laboratory and is now moving to lead her own laboratory at MD Anderson. Katie Taylor and Tara Barron, who are both presently postdoctoral fellows with me, are critically important collaborators, our funding sources, and lab members, past and present. Thank you so much for your time and attention.